very much for coming. There's a lot of folks here. I appreciate you making the trek over. It's good to see you all. So my name is Zac Forsman. I'm a researcher at Hawaii Institute of Marine Biology, which is this wonderful little island, Coconut Island. We take a boat ride to work every day. It's a state-of-the-art marine lab located right next to the coral reefs. So we have these amazing facilities where we can go in right off the reef and look at the genome of corals. We can do, we have this amazing microscope that's this in Ruth Gates's lab. This is an example of one of the images from this microscope that allows us to take an image of the live coral. So this is a single individual coral polyp. This is only 1 millimeter across. And all those little red dots that you're seeing are all the algae that live inside the coral tissue. So the corals are solar powered. They also are little mouths that feed on plankton. And there's all kinds of little critters that crawl around on their surface. So you can see some of these little dots moving around up here in the corner. These are unknown little eukaryotic organisms all over the surface. They're kind of like aliens from another planet. And I'm kind of a science fiction geek, and so this is something that to me is just like the closest I can get to studying something that's from another world. But if we zoom in at coral and take a closer look at corals, this is basically what their anatomy looks like. They are like an upside down jellyfish that secretes calcium carbonate, which is very, very similar to human bone. And that algae is inside the tissue of their tentacles. So they're in the phylum Cnidaria along with jellyfish, and what really defines that phylum, that group of organisms, is that they have these stinging cells called nematocysts. So just like jellyfish can sting you, corals can sting little tiny plankton, and some corals can actually irritate humans. So anyway, that's basic coral biology. This is what a coral skeleton looks like if you take away the thin living layer. So they kind of look like fingerprints or snowflakes. Each one is kind of unique. This is another example of the coral microscope in Ruth Gates's lab. So this is a close-up of a little organism. We're not even sure what it is, and we don't even know what its relationship is to the coral. This large thing you see right here is a tentacle. It's loaded with nematocysts, and it's loaded with all these little symbiodinium, the zooxanthellae, that live inside the coral. So there's a lot of unexplored territory. It's kind of like a new frontier. It's also, coral is a foundational species, which means it's kind of like the trees for the forest. It really forms the structure, the habitat for all the different organisms that live on coral reefs. There's a very interesting project done by the Smithsonian called the One Cubic Foot project, where they take a cubic foot and look at all the organisms within a day that pass through that cubic foot. There's this incredible amount of diversity. Up to 1/4 of marine life the diversity of marine life is found in coral reefs, even though coral reefs only occupy 1% of the oceans. So they're very important foundational species. And because there's all this incredible diversity, they're kind of the rainforests of the sea, you see a lot of interactions between these organisms. And this can result in kind of a death spiral of decline. So if you have increased sedimentation, you have less coral, you have less coral recruitment, you have more algae from more pollution, you have less fish recruitment from fishing, which results in less grazing, which also results in more algae. So you have this kind of death spiral, which we've seen occur especially in the Caribbean. So the coral cover in the Caribbean has really gone down at an alarming rate over the last decade or so. On the opposite end, we have a positive feedback loop. So it's kind of a boom or a bust ecosystem where you can have increased habitat leading to increased fish recruitment, increased grazing of algae. Decreased algae means more coral recruitment, which makes more structure, and so on and so forth. So how do we go from a virtuous cycle, or how do we get it into that virtuous zone versus the death spiral? And that's really what motivates my research interests. As you may have heard, corals have a lot of threats, both globally and locally, and this has resulted in a lot of decline. They really inform a lot, or they perform a lot of important functions, including nurseries for fish, for biodiversity, for shoreline protection. And we want to try to do what we can to try to slow or reverse the decline as much as possible. So my research has really kind of been to borrow from what we do on land. Human beings, we modify ecosystems, we plant a lot of trees, we develop nurseries, we farm, we do agriculture. We've really modified a lot of the planet. We really haven't done too much in the ocean except for maybe some aquaculture at small scale. So corals are very similar to trees in that they have lots of small offspring, and those small offspring have high mortality. So they don't really live that well. And so we make nurseries for trees to increase their survivorship, and that results in entire forests. So my research has really been focused on, can we do that with corals? Can we bend this mortality curve? So I started at the Waikiki Aquarium, building a coral nursery in the back of the Waikiki Aquarium. Really, my research was focused at the time on looking at how do we reduce the mortality from these small, on these very, very tiny colonies. So I would fragment the corals, and then I would take photos and basically watch them grow. So this is every 2 weeks. And I noticed that they really spread pretty rapidly. Kind of some of them would grow surprisingly rapidly, and it really depends on the immediate surroundings. So I set up 2 nurseries, one where I cleaned the corals with a toothbrush out in the hot sun. Drove me a little crazy. And then the other nursery, I hired some sea urchins. They work for very cheap, but they don't do as good of a job. You can see here's a little coral riding on the back of a sea urchin. So there's a very clear pattern where the sea urchins resulted in a lot more dead and damaged corals than the poor postdoc. We also did some very simple studies, just garden variety studies, looking at what species of corals do they respond well to feeds. So these are commercially available feeds that are available to the hobbyist industry. So there are a lot of people on the mainland that can grow reef tanks as a hobby. That's not allowed in Hawaii. Corals are protected here. So some very simple garden variety experiments. And while I was doing this work, I noticed that coral colonies from the same colony would tend to fuse together, and they do so in a really rapid way. And so I did a simple experiment just to say, well, what if we could exploit this to cover a really large surface area? So I took just a Home Depot bathroom tile and glued some coral on it. And in 38 days, 125 days, in about a year, the corals covered the complete thing, and we ended up with a coral colony that would take a long time to grow that large in nature. So something about this process really stimulated the wound healing and growth process, and there are very few competitors, so it kind of jump-started the growth of the coral. So this is what the colony looks like today. It's very natural looking. Here's with another coral species. There's the same colony today. And at the time, I was asked to work with U.S. Fish and Wildlife on this large ship grounding. And at the time, this was the largest ship grounding, largest natural resource damage case to a coral reef in U.S. maritime history. So there's 20 acres of coral that were damaged here. And part of the process is you have to try to figure out how can you do something at that site to make it better. So one of the ideas was to, based on that concept, to start a coral nursery. So for the past 3 years, and then of course another ship grounding happened. This time it was the Navy. So we built a coral nursery in order to explore this concept further and to look to see, can we mass produce corals? Does this nursery concept work? So the nursery consists of these large outdoor tanks. We have surge tanks that give a big burst of water flow. We have an indoor controlled facility where we can really tightly control the chemical and lighting conditions. And we began to mass produce onto these tiles. And we published a paper with Mote Marine Lab in Florida. They discovered the same technique, and so we co-published a paper. And this is a method that's working well for them. So it works on a wide variety of species. This is usually for the species that are slower growing, not the big branching species that are grow really quickly. So that's just some details on the growth. The growth rates are really, really surprisingly high. Just to give you an example, here I started with a piece of coral that's about the size of my hand, and it took me an hour to glue them onto all of these pieces of dead coral. This is all beach rock that washed up on the beach. And about 15 months later, each of these is larger than the starting colony, which is a pretty good deal. So if you get the conditions right, the corals really can grow surprisingly fast. So here's another example with another species. So that was really what was the idea, the impetus for starting this coral nursery. And we did experiments with tiles, and then we would connect the tiles onto these larger modules. And these corals actually reproduced. They spawned after they've gone through this process. So corals, they release their eggs and their sperm into the water column, and then those eggs and sperm meet together and make a little larvae, and the larvae settle. So maybe we're onto something to really jumpstart the coral growth, but we really need to figure out a way to do it at a larger scale, to scale up like a factory. So one idea is to scale up to borrow from what nature does. And one thing that you can find in a lot of environments is these little balls of coral where they're covered by coral on all sides. You turn them over, they roll around in the surf until they get wedged into a crack, and then they grow and attach by themselves. And if we could mimic something like this, it would really reduce the cost, because the divers in the water, it's very expensive to have divers in the water. The more time you have planting coral, the more expensive it is. So if we could develop a method like this and mass produce coral seedlings, we might be onto something. So I started making little designs out of clay, and I started working with local school groups, and they would come up with different designs, and they'd fire them in the kiln, and we'd try growing them, and they would grow over and fuse and then get larger and larger. So the idea seems to have some potential. This is what they look like after about a year. So they start to grow coral on all sides, and they begin to attach to whatever you set them down on, as long as they don't roll around too much. There's an international organization, a nonprofit called Secor, and they came up with the same idea. Secor wants to harvest those gametes, the larvae. There are thousands and thousands of larvae, an unlimited supply, but they have a very high mortality rate. And so their idea is, if you could get those larvae to settle on these little seedlings, you could mass produce them, and you could sprinkle them out, and you could really, over the scale of decades, you could grow a whole new reef. So that's just some of the work that's kind of going on in parallel. Lots of folks have kind of great ideas at the same time. Another idea that I have, which is what I'm funded on right now, is to try to develop a coral assay. So this is something that we could actually outplant. We could put this assay out and try to figure out what is killing a coral at a particular location and why. So, for example, I was involved with a big beach sand replenishment. Yeah, do you have a question?
Yeah, previous. The company that's harvesting larvae, have they had any success?
Yes, they have. But so far, there's only been one coral colony out of all of their trials that's managed to grow all the way up to adulthood and release eggs and sperm again. So it's still, there's a lot of science that needs to be done to understand what's causing the mortality of small colonies. And that's one of the ideas behind this growth assay, is to try to figure out, to narrow in those parameters. So we would have something like a data logger, sediment traps, and various coral tissue, and we could use that as an assay. So in order to get at that, I've been doing some pilot studies at HIMB, and this is just an example of one of the studies where we found that the corals really grow differently in different environments. They have very different survivorship. And some of it is unexpected. We've seen a lot of growth in this lagoon, which is really a place where there's no coral. It's a terrible habitat for corals. But these genotypes that survived in this lagoon in only a little bit of water, they survived 2 bleaching events. So there's something about it that seems to be hardy in these very bizarre environments. So in order to explore these ideas further and to develop, yes, you have a question?
Sorry, what is HIMB?
Hawaii Institute of Marine Biology. Yeah, sorry about that. Yeah. Yeah, so to explore these concepts further and also to develop a platform in order to do this kind of research, I came across an opportunity about last year where there are these former dolphin pins that they are no longer in need of. And there is these fish pins that they're also, the project is finished. And so this is what these abandoned aquaculture fish pens looked like, kind of a mess. But there were these corals that were growing on the nets. And so I thought, well, maybe we could just repurpose this and create a platform to grow corals. So with the help of, and so this is what it looks like on the fish pens. And this is what it looked like in the dolphin pens. These massive dolphin pens had these corals all over the bottom. Many of them were the size of dinner plates. And so with the help of lots of volunteers, and there's me out there wrangling these big plastic pens and pulling out old lumber and doing lots of manual labor. And then we created these platforms to grow the coral on. This is just a little video of what it looks like. So this is some of the corals we left out as a test for 1 year to see how they would do, and they did pretty well. It's just an example of some of the corals that are out there, different nets. So then we transplanted all of the corals on the outside and the inside of the nets with the help of all the volunteers and with the Division of Aquatic Resources. Here's, this is what we came up with as a pickup truck. So here we have a guy on a paddleboard and he's pulling this rack of coral behind him. So that's the ocean equivalent of a pickup truck. So we salvaged all the corals from these dolphin pens and from these old fish pens. This is what the environment looks like. We took out all the old structures that were rotting and falling apart. This is a new one that had a net. We had to cinch up the net so it wouldn't run on the reef and then move it over. This is a little time lapse of us moving the whole structure over, which is quite an adventure. So this is a salvage crew that was tasked with tearing these dolphin pens down, but we recycled as much as we could. This was right by the entrance, so from the lighthouse, right by Lilipuna Pier, you can see all the dolphin pins, but they're all gone now. So now they're off to the side. So our major construction is complete. We have this research platform, and we can do a wide variety of experiments on them. We have hundreds of coral to work with, and once they're already collected, we don't need a secondary permits to work on them from the state. So the state has been working with us. We're interested in transplanting these corals back in the environment, doing controlled plots to see what we can do to increase of that kind of positive feedback loops that I was talking about earlier. So those are the types of experiments that we'd like to do. This is just showing the beautiful area where the nursery is at, and this is what it looks like today. We've had, and this is a little time-lapse video of all the little baby fish that are starting to recruit and call this place home. And so the fish are coming in and they're feeding on the algae. They have a new home to come in, and there's a lot of manipulations that we can do looking at interactions between the fish, the coral, the sponges, and all the little critters that live inside the corals. We can do direct manipulations and direct instrumentation right on the reef in a pseudo-natural environment. So we have a lot of projects going on. We have a lot of students and interns that come through, especially in the summer. So these were all NOAA interns. This was a master's student from London working on coral assays. So lots of fun times, keeps me busy. And as if I'm not busy enough, that's actually kind of what I do for a hobby, for fun. My real job, or my real training, my real interest is in conservation genetics. And so this is really a whole other area of study. So conservation genetics is really using DNA as a tool. It's kind of like, if you look at this picture of the tree of life, this little geeky guy with the magnifying glass, that's me. Well, it's not really me. I don't have a beard, but
He's got more hair than you got.
he has way more hair than I do. That's true. So, but what he's doing is what I do. He's looking back at the tree of life to understand, what is it? Where did it come from? How is it related? How rare is it? If it's really, really rare, is it in danger of extinction? What can we do to preserve that coral, or that particular organism that may be rare? Does it have low genetic diversity? Can we prevent extinction or loss of biodiversity? Those are some of the questions that conservation genetics asks. Now with coral, it's a really important tool because corals are very, very difficult to identify. So this picture is really an example of variability within a species. So this is phenotypic plasticity. This is kind of like with human beings, some of us are able to suntan. So we can go out and in different environments, we have a different phenotype. We have a different appearance. So this is the variability within a species. This is the variability between a species. So it's very, very hard to identify some of these things. So we need DNA to kind of help us out. These corals can be very plastic. So for example, here's a coral that I was growing in culture, and it looks like one species, but it's starting to grow like another species after it's in a different environment than it's used to. So there have been a lot of studies that transplant corals, and they show all of these responses to different light conditions, to different water motion, sedimentation, interactions between species. There was even one study where they took corals and put them in a centrifuge and found that they grew differently under different gravity conditions, which is the coolest study ever. So what is the genetic data telling us so far? This is, I'll walk you through this really colorful diagram. Basically, each one of these circles represents a single DNA type, a DNA sequence. The size of the circle represents how many individuals had that type of sequence, had that genetic signature. The little links between represents the number of mutational steps. So this is like 5 or 10 different mutations away from this other cluster. And the colors represent where in the world these sequences were found. So the important thing here is that we have a couple of these circles that were only found in Hawaii. Like this blue circle, we only found that genetic type in Hawaii. We haven't seen it anywhere else. That means that it's not interbreeding all over the world. It's unique to Hawaii. But it didn't correspond to any of the named species. It had a wide range. So all of these different coral colonies that you see here were associated with that single DNA sequence. So what do we call it? A lot of the experts disagree about some of the names. It's very, very challenging. But we know that we have something that's unique here in Hawaii that doesn't occur anywhere else. There's also something unique in French Polynesia and Moorea. So here's an example of another example of a genetic tree. So this is really just showing you these branch lengths are telling you how genetically different things are. So this group, this type 1 group, you can see all of these different coral colonies, which we were hoping originally we wanted to name different species, that they're new species, but it turns out they're all lumped together. But if we look under the microscope, what we found was that this group seemed to have this trait that was distinguishing. So there's something that's agreeing with this one genetic marker. But one thing that we didn't know is that's only one gene. What about if that one gene is under selective pressure? What if the corals are interbreeding and the genes get all messed up? So we needed to look at genes across the entire genome. Now, recently there's been a big advance in biotechnology. So for the same effort a few years ago that we could sequence a single gene, now we're able to sequence entire genomes. It's a flood of data. It's so much DNA data. Just to give you an idea, when I graduated with my dissertation, the study that I did could fit on 1 page of text. That's how much data. So it's maybe a few kilobytes of data. Now we're talking about terabytes of data, whole hard drives full of data. So it's like drinking from a fire hose. How do you identify all this stuff? It all becomes computer science very, very quickly. So I do most of my time in front of the computer, and then I need to take a break from that, and that's why I do the Coral Nursery stuff. So here's an example of basically how that works. I won't go into the details because I don't want to bore you. But this is an example of the entire mitochondrial genome. So that's 18,000 base pairs, which is the letters of DNA. And long story short, all of that agrees with, it tells us the same result, that all of these colonies really, all of the coral colonies is very plastic. It's very flexible. And the reason why, we believe, is because it's all happened very recently. So if we assume a molecular clock, we can actually go back and put dates on when these different corals diverged. And we think that this group evolved very, very recently, about 3 million years ago. And some of the corals, so this is if you look at the individual genes, you can actually see how they exchange genes. So some of the more recent corals may actually hybridize and interbreed, but most of them don't seem to. So it's not a story of interbreeding and hybridization. It's more a story of recent evolution and plasticity. So here's another example. This was stemmed out of my dissertation work. So this is a Loeb coral. It's very common here in Hanauma Bay, one of the most common corals in the Pacific. So here we have these two different species right next to each other. They look very, very different. This is another genetic tree. Long story short, we have groups where we have things that look very, very similar, but genetically they're very, very different. We have a few cryptic species, these little hidden things that we didn't know were there before. They're little tiny things that kind of everybody would ignore, but sure enough, they were different. So this was my first experience with describing a new species, which is quite an honor. It was named after Dick Randall, a coral biologist. And we discovered another little cryptic species here in Honolulu Harbor, this little tiny guy that's one of the smallest corals. It reproduces crawling larvae, and it, instead of growing big, it just reproduces a lot. So it puts all its energy into reproduction instead of growth. But the coolest thing was that we had both these things that we thought were different species, and genetically they were hard to tell apart. Same in another group, and yet in another group. What's going on here? And this is the original type specimens from the Bishop Museum here in Honolulu. And so what it looks like is that there's kind of a gradient going from one species into another, from the branching to mounding form. So is this a case of hybridization and interbreeding? In order to look at this, we used that next generation sequencing method, and we found that within this group, there's a lot of interbreeding. There's a lot of kind of genes that are mixing around between these different corals with the branches and the mounding. So this is some brand new data. And what we're looking at is one group that seems to be mostly this branching coral, one group that's mostly the mounding coral, and then an intermediate group that has a lot of mixture of both. We've also been discovering all of this really interesting genetic structure by habitat. So the corals really seem to be not interbreeding that much between different habitats. So if we look at the inshore area versus the offshore area, there seems to be a lot of genetic structure. So this kind of work can inform coral restoration work, because you wouldn't want to transplant or move corals into a place where they wouldn't survive. So it teaches you a little bit about what corals grow where and why. So the two kind of fields that I'm involved in that are very different and pull me in different directions, they actually do kind of relate to each other somehow, or at least that's what I keep telling myself. We've also done some work on some very deep water corals. So these are really interesting corals that occur in the mesophotic habitat. So the mesophotic habitat, this is area of deep water reef. So this is somewhere in the neighborhood of 60 meters. These corals actually dominate all the way down to 100 meters. So that depth, that's like 300 feet. This is the area where it's perpetual twilight. But corals, they're solar powered. So somehow there's algae that live in the tissues of these corals that enables them to survive and be the dominant organism at this deep depth. So they're very specialized. So in this genetic study, that's what we found, is that some of these corals were very unique to a certain depth. They're very specialized. The Symbiodinium, the algae that lives within their tissues, this is looking across a depth gradient. And we found that some of these algae only occur at very deep depths, so 95 meters and below. This is an image of what it looks like down there at these depths. So amazing kind of fields of these whirling coral colonies and a diverse community that they support. If we look at the coral DNA and we line that up with the Symbiodinium DNA, the algae DNA, there seems to be some coevolution. It looks like they both kind of develop that specialization to deep water together. But there are some cases where the DNA actually touches on conservation directly. And so for this example, this was the rarest coral in the world. And DNA actually found that this coral was not really that likely to be rare. There were only 5 colonies that were ever found, and they're all found downstream from the opening of the Isthmus of Panama. The coral looked a little bit different than the things in the Caribbean, but according to the DNA, they're identical. And so that was my very first paper, and I felt really bad about it because it was basically killing an endangered species. It's saying that this species is not really separate. It might not warrant protection. And to make matters worse, this species was also named after my advisor's colleague, so I just felt awful. And the results were also, it was only one gene, one marker, and so I didn't really have a lot of confidence to know whether or not this was a correct result. Am I doing the right thing? But luckily, more recent studies have backed this up. And then Peter Glenn himself, who the coral was named after, he actually solved the mystery and found out that what happened was there were researchers that moved some corals from one side to the other at exactly that location to do a study. They thought it was dead. They didn't know that there were still living corals, and they sprouted back again. So, yeah, killed an endangered coral. Sorry about that. But it's better not to waste resources, I suppose. But here's another case that's a little bit more challenging to deal with. So this is a very rare coral that occurs in Kaneohe Bay. There's only maybe half a dozen to a dozen colonies that are known to exist. This is what they look like, beautiful coral. Here they are. This is Kaneohe Bay. There's Hawaii Institute of Marine Biology, Coconut Island, right there where I work. So in this case, the DNA really lumped everything together and said that it's not really that different. And so it really prevented this coral from being listed under the Endangered Species Act. Now, in order to really know if it's a different species, we need to cultivate it and do breeding experiments to know whether or not it interbreeds with some of the more similar species. So this is showing some of the corals in that species complex. They look quite different. They have a range of morphology. If you zoom in under the microscope, they all kind of have similar characteristics and look quite alike. So we went through and we measured a bunch of things, and the long story short, all the measurements we did of the microstructure also agreed with the genetics together. But this coral is really known, this genus is really known to be very variable. So this is an example of, this is all considered one species. This is this rice coral, Montipora capitata, that grows in Kaneohe Bay and here in Hanauma Bay. And you can see it has really diverse growth forms, all one species. And so one of the experiments that I did was to transplant these into different environments. So we have different genotypes. Each one of these numbers is the same genotype after 1 year of growth in these different environments. And what you can see is that there's more variability by the environment than by the genotype, which really, you can still see some differences between individuals within the locations. That's called a common garden experiment. But really, most of the differences are between the different environments. There's a lot of plasticity. And here's another kind of cool example of some of the different variability within a species. So this is if you take an ultraviolet light and you shine it on these two coral colonies, which look very, very similar under the same lighting conditions. If you shine a UV light on them, they autofluoresce. They're like a black light poster and kind of glow in the dark, and they look really, really cool. But you'll see big differences between individuals, and we don't yet know why. Is this a stress response? Is this because they're adapted to different habitats? It's still kind of a mystery, and this is another thing that cultivating and growing the corals, we can start to kind of unravel some of these secrets. But I also wanted to say that in the case of this this really rare coral, this Montipora dilatata in Kaneohe Bay, this paper that I wrote prevented its listing under the Endangered Species Act, because in order to protect it according to the Endangered Species Act, it has to be a separate species. This was taken in 2015, so this is one of the beautiful coral colonies that I'd worked with for years. There were some major global bleaching events in 2014. We had another global bleaching event in 2015. It's been one after another, and this is really unprecedented in Hawaii. It's not really happened before. We had one bleaching event back in 1996 that wasn't anywhere near as severe, but now we're starting to see bleaching events year after year. We've seen some major mortality on the Big Island and on Maui. And so this was really something that kind of hit home to me, that was a little bit worrying, and I felt really, really bad that that paper kind of prevented the listing of this coral. But nevertheless, that's what the data says, and we still don't know. We're still working on trying to figure that out, and we're also trying to figure out how to cultivate this coral because it is unique. So, kind of as a quick summary, am I way fast on time? I think I kind of buzzed through a lot of that. But in just a quick summary, my work is really interested in kind of focusing on where you can prevent the decline, these negative feedback loops. But I think more importantly on what can we do to kind of, for these positive feedback loops, how can we really interject more coral habitat? How can we get this cycle to roll into a positive direction? These are some of the great people I get to work with. So I work with Rob Tonin and Brian Bowen. They're the two PIs that run the genetics laboratory that I'm in. Rob studies everything without a spine. So everything in the ocean that is an invertebrate, all the animals anyway, so about 22 animal phyla. And then Brian studies all the charismatic, interesting megafauna like fish and dolphins and monk seals and all of that genetics. Great group of people. So these are all the postdocs in the lab. So we're the folks with PhDs that don't have the cool jobs yet. I've been surviving on soft money, which means I have to write grants like every year to keep myself afloat. But I love it. I get to do the coolest projects in the world. These are all the graduate students, so aspiring folks. They get to go all over the world. They get to travel all over the world and do really cool research, including a lot of rebreather research. So this is into some of this mesophotic habitat, which is human beings have only recently begun to explore, and we're finding all kinds of new and interesting species in these unexplored habitats. So it's quite a privilege to work with those folks. The Poly Foundation and the Seaver Institute have funded my work, as well as all of these government organizations from time to time. So I'm always working to try to find grants to support myself and students. This is the team at the Division of Aquatic Resources that built the Coral Nursery that I was talking about. Really so many people that I have to thank for all the work that I've been involved in. It gets kind of dizzying. And then I have all the students. So these are students and volunteers, people that help me out in the field, and usually it's part of a thesis project or something like that. So they come for a summer experience and they have to write a thesis, or they're master's students, and now I'm working my way up to PhD students. So these are multiple year, larger scale projects. So that's just an idea of the type of fun that we have out at HIMB, and all the great people that we get to work with. And I'll finish early and take questions. Yes, sir.
A question about corals, not what you were talking about. Answer to how does one coral communicate with neighbor when it's spawning? There seems to be some sort of communication.
That's a very
so when there's two different species get close to each other and they're warring each other.
Yeah.
There's some communication. Is it in the water, by contact, chemical, what?
Okay, so there's two questions here. The first question was, how does a polyp within a single coral colony communicate with others in order to know, like, for example, when to spawn? So I'll try my best to address that one first. So the corals are all, each one is kind of like a little upside down jellyfish, but they have these little channels where they share resources. They pump some of those resources back and forth together to each other, nutrients and whatnot. It's, well, let's see.
I was going to say mucus, but it isn't.
It's not the, mesoglia are the different tissue layers, but there are little channels that go in between the mesoglia. So it's kind of a network of little, it's basically the corals just have two different tissue layers, and there's kind of a lot of area for fluid to go in between. How they coordinate together to spawn, though, there's mass spawning. And the corals actually have they have the ability to sense changes in light, period. So they're actually genes that code for like little, not quite eyes, but they can sense light and dark. They also have temperature cues that let them know when it's time to go off. And then there are, in the ocean, there's a lot of hormones that are released. So once one coral starts to go, then all the other ones are like, hey, now would be a good time to reproduce. So they kind of put all of their eggs into one basket, so to speak. Let's see, and then the second part of your question.
When the two corals get close and they want to...
When two corals get close, when they compete for space, so they actually, many corals can actually turn their guts inside out and attack each other with their guts. So some of them have stinging nematocyst cells, and some colonies actually specialize in parts of the colony where they'll have parts of the colony that have more cells than others. And those are like the warrior polyps, and they'll fight with the other colonies. So it's kind of like a super organismal type of a thing. They can actually extrude their mesenteries and chemically sense each other. There's a lot of chemical warfare that goes on too. We're starting to understand some of the chemical compounds that some of the corals exude, just like plants do.
One edge is doing the fighting. Does the rest of the colony have anything to do?
Well, so the whole colony can actually die if it's losing the war. They'll shift all the resources into the part of the colony, and the whole thing will get weak and die. So yeah, some of them can actually pull out of their skeletons into little ciliated balls and go move somewhere else. So there's this thing called polyp bailout, where they're just like, we're out of here. So this very weedy species of coral can do that. And then they can also, some of the coral larvae are actually genetically identical, and so they can fuse together. So the same species, if you wound it, it'll release larvae that come back and heal it. or they settle together and make a colony really quickly. So corals they're just very bizarre organisms, very neat to study, and they always throw you for a loop. Unexpected things all the time.
Thank you very much.
Yeah. Yeah. Yes.
I understand that the mesophotic zone has the highest level of endemism amongst fish. And is that the same for corals? And if it is, why?
Okay, so the question is that the mesophotic zone seems to have one of the highest levels of endemism for fish, but is it also true for corals? And if so, why? So fish geneticists really have a huge advantage over the coral geneticists, and that is mainly that the mitochondrial genome evolves very quickly and very clock-like, and so we just sequence a single gene and we can really get a clear picture of what's going on. With corals, that doesn't work. So we have to do all this other genetics and look at a whole bunch of other genes. And then you also have this hybridization going on, and then you have the inability to identify corals in the first place. So I'd say we're just scratching the surface of what's down there. So some of the work that we've done is just saying, wow, there are these corals that are genetically distinct, but that's in Hawaii. We have colleagues in Australia, we have colleagues around the world, and we're trying to all of us work together to pull this information together to understand what occurs where and why. In some ways, it's kind of a race against time, though, because we don't even know what we're losing. So as to the part of your question about why are they endemic at those levels, it's really a puzzling and interesting question, because a lot of the fish in the photic zone, in the shallow parts, some of them are endemic, but not all. There's some that have very, very broad distributions. And it's not really clear why, but one hypothesis is that it has to do with stability and with being in a place for a very long period of time. You start to form a niche, and then you outcompete anybody else that tries to come in. So you're there for a longer time in a stable environment, whereas with sea level changes, with glaciation, the shallows change every 100,000 years or 10,000 year cycles, and so that habitat is constantly in a state of flux. So that's one hypothesis, but those are the kinds of things that Brian Bowen in my lab is really interested in looking at with and using fish to test it. With corals, we're way far behind.
How soon can you hurry up and start replanting the coral reefs that are dying?
How soon can we start replanting coral reefs that are dying? I would love to be able to say that we've figured it all out and that we're mass producing and that we've got factories and we can go. But I think we're really in our infancy, especially compared to what people do with forests. And we're also, there's another kind of component to this, which is we also need to simultaneously address what's killing things at the same time. If we go out and plant a whole bunch of things and then it all dies, that wouldn't be a good thing. So we're really at our very beginning steps of kind of understanding this ecosystem. It's also a hurricane, so if you ever try to do anything underwater, if you ever try to construct anything underwater or do anything, it's comical. It's ridiculous. You get all scraped up, you get bumped around. So it's been something that it's a slow process, and we're trying. There has been major strides in the Caribbean, though, because they're dealing with their major coral that's really declined is also an endangered species. So they've been doing these huge efforts involving a lot of volunteers and divers, and it's been making a big difference. So they're outplanting these corals, and whole reefs are coming back. Those corals are a little bit faster growing, so they have a lot more advantages than we have, and they have a lot more experience. Here in Hawaii, we're catching up a little bit with all the lessons that are learned in the Caribbean. But it is making a big positive difference in the Caribbean, and it's really turned from, this whole field has been something that was kind of snake oil salesman in the beginning, and now it's turning into something that's more legitimate. More and more scientists are really realize has value because of the work that's happened in the Caribbean. So back in the back.
So when I go snorkeling and diving whenever up here, and I may see something that I can congregate my steak on, and then I see something right next to it, let's say green coral. there's very good likelihood it's comes from the same genotype.
Question is, if I'm swimming along and I see two things that look really, really different, are they really different, or could they be the same genotype? So in the case of elkhorn coral and brain coral, those are way different genera. So that would, those are, if they're really, really, really distinct at multiple levels. So if you look under the microscope and they're really distinct, then you look at the, then they're probably different. But corals are modular. And so maybe they can just rebuild the way that they grow together slightly, and that changes the coral, the shape of the coral. So we know from all the studies when we transplant them that they can do that. And it's kind of like, you know, they're stuck in a habitat in a certain environment, and so they can't really. It's very hard for them to move. You know, that's the only way to cope with it.
So you were talking about hard corals. What about little animals to the soft corals? Do you have any example of soft corals?
Yes. So the question is, I was talking about hard corals. Is there similar work going on with soft corals? Yes. And so we have a lot of the same issues, the people that work on soft corals and hard corals. So we collaborate with each other a lot, and we have a lot of sympathy for each other. So yes, there's work that's going on in that direction. More? We have time for more?
Yeah.
Okay.
Looking toward the future, do you find much pressure on you to look for corals that would survive in warmer, more acidic water?
Yeah, so the question is, do we find a lot of pressure to find corals that would survive in warmer, more acidic water? And the answer is yes. So especially in the Caribbean, this is an area where they're doing a lot of nursery work, they're doing a lot of outplanting. And what they're finding is that some corals are much more tolerant of warmer water conditions, but usually that comes at a trade-off. Usually if they're more tolerant of the warmer conditions, they don't do that well when it gets too cold. So it seems like they're really uniquely adapted to that habitat. And as that habitat is changing, we're definitely, you want to know which corals go in the right environment to know a little bit about what you're outplanting.